Hello, good morning. Thank you for being on time. We will go ahead and get started. I want to do a quick recap of some of the key concepts that we covered last class before we continue with the new material. So if you could please take out your study guide for lecture 5, we would fill in a few questions from there to start. So I'll give you a moment to get that ready. Okay, so here the question asks for the word equation as well as the chemical equation for cellular respiration. And you should be familiar with that by now. We have glucose reacting with oxygen. And then it is going to become carbon dioxide plus water. And in the process, we are going to generate energy in the form of ATP. On the other hand, for the chemical equation, we have C6H12O6 for glucose plus O2, which then produces carbon dioxide plus water and 36 ATP under ideal condition, right? And we have to balance this. So we got to put the 6 and the 6 and the 6 here for all of these reactants. Now, we also learned that this is a redox reaction, right? In a redox reaction, one reactant is going to be losing electron, and then the other one is going to be gaining electron. When you lose electron, we call it oxidation. And when a molecule gains electron, then we say reduction. So can someone please tell me, is glucose oxidized or reduced in this reaction?
Oxidized?
Sorry, is it oxidized or reduced? I didn't quite catch that.
Oxidized.
Oxidized, that's right. So we are going to be removing electrons from glucose. And when we have finished doing that, it eventually becomes carbon dioxide, right? Okay, so this is oxidized. And where do those electrons go? Ultimately, the electron will be transferred onto oxygen. When oxygen picks up those electrons, it becomes water. So this is reduced into water, right? Because the electrons are ultimately going to end up on the oxygen. Sometimes we call oxygen the final electron acceptor. Right? We'll come back to that point later, but I just want to get that in your head now, right? Because oxygen is the ultimate molecule that will be receiving those electrons. We call it the final electron acceptor. Okay, so nothing is new. We're just reviewing for those of you who are joining us. There are 4 stages in cellular respiration. The first stage, as you might recall, is called glycolysis. And where does glycolysis take place?
In cytoplasm?
Cytoplasm, that's correct. Thank you. Now, the key products that we get from glycolysis include 2 ATP. And I want to remind you, this is net because we invested 2 ATPs, right? In energy investing phase. And then in the yielding phase, we get 4, which is why net is just 2. 4 minus 2 gives you 2, right? In addition, we also get 2 NADH as well as 2 pyruvate. And at this point, most of the energy is still within the pyruvate molecule. Okay? We can still take the pyruvate to the next two stages where you will be able to create more ATP, more energy carrier. Okay, now I want to put a star here. We talked about how there are two methods for producing ATP, right? One of them is substrate level phosphorylation, or SLP for short. That's when there is an enzyme that transfers a phosphate from a donor molecule onto ADP. Is that the common method of making ATP, or is that the less common method?
Less common.
Less common.
That's right. Okay, that is the less common method, right? Only about 11% of ATPs are produced this way. These two ATP that we get from glycolysis, that is from SLP. Okay. Next, we have pyruvate oxidation. Excuse me, pyruvate oxidation. That happens in the matrix, which is the innermost portion of the mitochondria. The key products of pyruvate oxidation include 2 carbon dioxide, 2 NADH, and 2 acetyl CoA. Right? All this is per glucose, right? I'm going to put here per glucose. For every glucose we break down, we end up getting 2 pyruvate. Each pyruvate will undergo 3 steps within pyruvate oxidation, right? First step, removing a carbon dioxide. We call that decarboxylation, right? We wrote it down last time. Second, we're gonna release high energy electrons, which will be captured by NAD to create NADH. And by this time, I keep on saying, the molecule is not as energetic anymore, and therefore we need to recharge it a little bit. We really need to give it a boost. So we add a special molecule called the coenzyme A, and afterwards we get acetyl-CoA. All right, no ATP is produced directly in this stage. Then we have the next cycle, next stage, which is called the Krebs cycle. And the first product in the Krebs cycle is citric acid, a 6-carbon molecule, which is why sometimes this is also called the citric acid cycle. It also takes place in the matrix. Now, why don't you take a moment and look at the key products for the Krebs cycle?
I would say it's a 6 NADH, 2 FADH, 4 CO2, and 2 ATP.
That's right. Okay, 6 NADH, 2 FADH2, 4 carbon dioxide, and 2 ATP, right? 2 ATP. So again, I'm put a star here. Those ATPs that we get from substrate level phosphorylation. Okay? Does anybody have any question about any of these things so far? Again, we're just reviewing, nothing new at this moment. Anything at all? I'm just opening a file while I wait.
So there's no ATP in pyruvate oxidation. It's just in glycolysis and the citric acid.
That's right. There's no direct ATP production. Right? There's like you're not making any of it. Even though we generate NADH, which will eventually be used to make ATP, right? So that's why I keep on saying it's not like direct ATP production, right? You will indirectly make ATP. Does that make any sense?
All right. Thank you.
Yeah. All right. We also talk about the difference between NADH and FADH2, right? Which one has more energy? Which one can make more ATP?
NADH.
That's
DH.
right. That's right. Each NADH carries enough energy to make 3 ATP, whereas each FADH2 can only carry enough energy to make 2 ATP, right? Having said that, let's try a few poll questions here and see how it goes. Go ahead, please. Okay, that's all for now. So first one, NAD plus has more chemical energy than NADH. NAD plus is the empty taxi, right? It hasn't picked up the passengers yet. It hasn't picked up the high energy electron. So it shouldn't have more chemical energy, right? It only has more energy after it picks up the electron, after it becomes NADH. So, excuse me, the first one is not right. B, NADH has more chemical energy than FADH2. I literally just told you that a minute ago, right? Each NADH can make 3 ATP, and each FADH2 can only make 2, right? That's why this is the true statement. C, NAD plus can be oxidized into NADH. In order to convert NAD plus into NADH, you need to reduce it, right? We wrote it down last time. Let me just find the notes right here. You just go back to the notes we took last time over here, right? NAD plus and FAD are reduced into NADH and FADH2. They are reduced because they're picking up electron, right? So it's important to go through your notes and see what's in it. And finally, glycolysis can still take place without NAD plus. Without the energy carrier, there's nothing to capture the electron, right? No one is picking up the passenger, right, so to speak, and everything is just going to stop. So that is not true. Okay, let's try another one here. We did this already, but let's try this again. I mean, this is different numbers, but go for it. Okay, so that has been a minute. This is 4 NADH, so 4 times 3, because each NADH can make 3, right? That's 12 ATP. 3 FADH2, 3 times 2 gives you 6, so total is 18. Okay, if you don't understand this, you should ask me a question. You probably will have to do calculations like these on the quiz, on the test, which is next week. All right. Now this one is a little bit tricky, okay? Give it a try. Okay, so thank you for those who have participated. If you're a little bit stuck, this is how you do it. Okay, one cycle. What are the key products of Krebs cycle if you spin it once? Look at the diagram we drew last time. All right, let me see if I can pull it up right here. Right? One cycle creates, this is one spin, right? 3 NADH, 1 FADH2, and 1 ATP. Right, so if we go back to here, we can write that down. 3 NADH, 1 FADH2, and 1 ATP. That's one cycle. When we fill in the table earlier, we put 6 NADH, 2 FADH2, and 2 ATP. That was for one glucose, right? For each glucose, you have to spin the cycle twice because you create 2 acetyl-CoA, right? So these NADH will go on to the ETC. Remember, they're just energy carrier. They will carry them to the final destination, right, to the ETC, where the high energy electrons will be used to make lots and lots of ATP. For each NADH, we can make 3 ATP. So that's 9. And then this is times 2. And then ATP is just going to be ATP. We add them up. 9 plus 2 plus 1 is 12 ATP. So one spin, one cycle will make a total of 12 ATP. If you were spinning it 5 times, then I multiply this by 5, right? Which means the answer is 60. Questions?
Hi, please can you explain it again?
Can I explain again? Which part?
Like the ATP getting the cycle and how you got the 12.
Okay, do you understand where these numbers come from?
Yes, I do.
Okay, do you understand why I multiplied by 3?
Yes.
Okay, and also you understand why I multiplied by 2, right?
Yes.
So 12 ATP, I just add them up. That's for one cycle of Krebs cycle, right? So the question said you turn it 5 times, right? You do the cycle, you spin it 5 times. So if each cycle produces 12 ATP, and I do it 5 times, then I multiply them. That's how I get the 60, right?
Yes, okay, I get that. Thank you.
Okay. All right. Anyone else? Okay. That's where we left off from last time. So today we will talk about the electron transport chain. Okay, the ETC, which happens in the cristae. That's the folding of the inner mitochondrial membrane. And then the key products would be 6-water. And we are going to get 32 ATPs. Okay. Remember, we for each glucose we will make up to 36 ATP, right? So far we only made 4. That's why we need to make 32 more. Okay. And you will also regenerate NAD plus and FAD. Okay, never mind the numbers here, like how many of each. You just have to know that they are recreated, right? And what will these do? These will go back, right? Go back to 1 to 3 to pick up more electron, right? Now, if you're still with me, and I hope you are, I'm gonna put a star here. Maybe I should choose a very different color. They look very similar. What other color do I have? Green, maybe? How are those ATP produced? Not substrate level phosphorylation. This one is oxidative, right? Phosphorylation. Okay, which I haven't explained to you how it happens. We're going to do that now. Okay. All right. I'm gonna take up the 2 questions on the test with you. Okay. So one of the questions, if you remember, everybody got the same question this time for the written response. One question was a hot environment, right? Hot environment. And we're looking for like membrane adaptation. This was the question that I put stars, right, on the PowerPoint slides. You remember I gave you guys like a breakout group. I let you guys discuss the question, and then we came back, and then I put 3 stars. I said I would know this if I were you, right? The only difference is the discussion you had in class was for cold. Exactly, right, Mona? That was cold. And a lot of people just copied the exact same thing and put it on the test. I bolded the word hot environment on the quiz. So almost half of the class put the answer for the cold environment version, okay? So for hot environment is very, very hot. So the membrane is going to be very fluid, very liquid. It's gonna melt, right? So the adaptation would be to counteract that. You want the membrane to be less fluid. You don't want to make it even more fluid because then everything would just leak out, right? So to make it less fluid in a hot environment, you need to increase saturated fat, right? Saturated phospholipid. Okay, you need to decrease unsaturated phospholipid. And as for cholesterol, do we want more cholesterol or less cholesterol?
Less.
That's right, because in the class, I wrote down more cholesterol means increased fluidity, right? So you need to decrease cholesterol, right? So what I was looking for is this hot environment. You need to tell me that it makes membrane more fluid, okay? And then you need to say adaptation should lower fluidity, right? To counteract that, right? Like if it's already fluid, you don't want to make it more fluid, right? Keep on saying. And then you need to write down these three things. So that was the 4 points. 1 mark for this and 1 mark for each of these things. Kushina, you have a question, please go ahead.
Yes, sir. I wanted to say about the quiz. It's like first the time, because if we have to read each question like 3 times, I don't, how much did we get? Something like 50
Yeah,
minutes?
50 minutes. Yeah, 50 minutes this time.
Yeah, I don't feel like it was like enough time to answer all of those because we have to read it. And I studied, like I read my notes, wrote them over, which is usually a good way to keep stuff in your remembrance. And then when I saw the test, I felt like most of the things that I studied like wasn't on the paper, or the questions seemed to be like worded in a complicated way. And then so it takes time to read it properly, make sure you're getting the understanding. So you probably spend like 2 to 3 minutes on a question for quite a few of them, if it's like 25 or 30 questions for 50 minutes. So
what would you
anxiety.
say is a reasonable time?
Probably 1.5 hours.
1.5 hours?
Yeah.
Oh, that would be way too
or
much.
Even
though.
1 hour and 10 minutes or something, because like it was like a lot of anxiety in a quiz. And I'm like, some questions I left it for like to come back, to go back over it. And by the time I got back to it, it was like 5 minutes left. I'm like going crazy. I left even the questions that you're going over right now. And I'm like, I don't remember what the heck I just, like it was a lot of anxiety for me.
All right.
Because of the time and how the questions were set up. It wasn't very straightforward. It was really challenging my brain.
Yep. Thank you for sharing that. And I think some people said I agree. So I think the sentiment is shared amongst your classmates. Yeah, sure. I'll take that into consideration. I'll lengthen the time for the test that's coming up. Hopefully that will help you a little bit. Okay. All right. The second question was mitochondria, right? Describe the relationship between the process that happens in mitochondria and chloroplast, right? Okay, and there was a diagram in the lecture notes that was that's important for this. Let me see, where was it? It's this one, right? That we spent quite a bit of time talking about it, right? So that's all I was looking for. Okay, it was 3 marks. So I wanted you to tell me that the mitochondria takes glucose and oxygen to produce carbon dioxide and water. That's 1 mark. And I want you to tell me that the chloroplast takes carbon dioxide, water, and convert it into glucose and oxygen. That's the 2nd mark. And the 3rd mark is to tell me that they are complementary processes, right? Meaning the reactants of chloroplast are the products of mitochondria, and the reactants for the mitochondria are the products of photosynthesis. Okay, that's what I was looking for. All right. So that is that. I mean, don't get too bothered by it. It's you know, not the end of the course. There's still a lot of grades for a lot of opportunity for grades. You know, for the lab reports, there are 2 lab reports. They're 10% each, right? You put in the work, you do it, you should be okay. I'm getting some emails from people asking me to explain what the lab is about. I'm not the one who's going to be marking it, so I shouldn't be the one who is explaining it to you, right? Professor Miran will take over, and when he does, he will tell you about it. But it's important. Like in the past, people lose marks for the lab primarily because they don't put in the work. Okay, like there's no other way to put it. Okay, like you have to watch a video for the lab. Almost all the answers are in the video. The video is just 8 minutes long, right? So you just have to watch it and literally just copy what the person is saying, right? For some of the stuff, you might have to phrase it a little bit differently to fit the question, but essentially, it's quite easy to get full marks, okay, on those things. Anything else you want to let me know? It's okay. Like if you have some complaints about the quiz, other things that you want me to know, you can tell me. It's this is a safe environment. I'm not gonna, you know, get upset or anything like that. Like so I heard that time is not enough. I will fix that. I heard that some questions are worded weirdly, but that's just the way it is, right? Like the questions are like it's not just my questions, right? We have several people teaching the course. We all came together and created questions, right? We felt that those are the questions that would be appropriate for the quiz. I can bring that up to them, but for now, nothing much I can do in that regard. Anything else, please feel free to let me know. I'll turn off the recording so you don't feel like you're being recorded. So the next thing we're going to learn is electron transport chain. Electron transport chain, the ETC, right? The final destination. We're going to draw a picture to explain this. I suggest you get a new piece of paper, and you should be doing it sideways, and you know, you should be drawing it like this. Okay, take up the whole page. So let's just make sure that you know where we are. We are going to take a mitochondria, and then we're going to take a look at this location. Okay, we're going to take a look at this location. We're going to zoom in. So here we have the outer mitochondrial membrane, and here we have the inner mitochondrial membrane. So let's just label that for you. Outer membrane, inner membrane. That forms the cristae, right? So out here is the cytoplasm. And then over here, that's the intermembrane space. And what's over here?
Matrix.
The matrix, that's right. That is the matrix. So within the inner membrane space, within the inner membrane rather, the cristae, we're gonna have these membrane proteins. Okay. they are a series of membrane proteins that are right next to each other. And that is what forms the electron transport chain, the ETC. Okay? ETC, ETC, ETC. And there are thousands of these ETCs in the crystal. That's why we wanted to fold up the inner membrane. The more folded up it is, the more space is available to pack these ETCs in. Okay. So we have the NADH that's coming from the second step or the third step from pyruvate oxidation from Krebs cycle, right? And this is what's going to happen. Take a look. The NADH will go to the first protein. These are proteins, okay? First protein of the ETC, and then it will deposit the electrons. Once it deposits the electron, what is it going to become? When NADH,
NAD plus.
NAD plus, that's right. It becomes the empty form again, right? It lets the electron off and it becomes NAD plus. Where is this NAD plus going to go? It's going to go back to the earlier stages, right? To bring more electrons to the ETC. All right. So what's going to happen to this electron? This electron is going to get passed down the ETC. Okay. It's going to get passed on to the next one and the next one and the next one. As the electron is being passed down, the energy from the electron is used to pump H plus from the matrix into the inner membrane space. Like this, right? So same thing over here. You're gonna maybe have another NADH over here, and then it's gonna pump the H plus. So over time, you will have lots of H plus in the intermembrane space. I'll give you a moment to catch up. Now you will also have some H plus here, right? Just not a lot because you keep on pumping it over, right? So I want to ask you, where does the H plus wants to go? If you leave them be, do they want to go from intermembrane space into matrix or matrix into intermembrane space? If we ignore all the ETCs and all the pumps for a moment, if you just leave them alone, which way does the H plus want to go?
From the
More
matrix to the
concentration.
intermembrane space.
Okay, and then what was the other answer? The Kira?
Where the more concentration is.
So does it want to go to matrix or does it want to go to intermembrane space?
Matrix.
Okay, let's do a poll. Let's see what people think. Okay. My question is, if you just look at the H plus right now, we have a lot of H plus in the intermembrane space. We have not a lot in the matrix. I'm asking you, which way will the H plus have a tendency to go towards? Is it A, the intermembrane space, or is it B, the matrix? Where does H plus want to go? Okay, so for those who have participated, we guess half here. Well, the answer is the matrix. Remember, things tend to go from high concentration to low concentration, right? So these H+, we have high concentration right now over here. High H+. Because the electron energy is used to pump them into the intermembrane space. And then in the matrix, we have low H+. Okay? So H+ have a tendency to want to come back into the matrix, but they cannot do it right now because the inner membrane is phospholipids, and that does not allow charged particle to go through, right? Okay, so even though they want to come back in, but they cannot. Okay, because the inner membrane would block them. So the only way to come back in is through a special protein. Okay, there is a special protein that looks like a doorknob, and this is called the ATPase or ATP synthase. Okay, and that allows these H+, these H+ to go back in like that. Okay, so this is like a dam. You store up the water and then you let it go through a turbine to generate electricity. That's how a water dam works, right? Similarly, we store up the H+ here in the intermembrane space, and when they go through the ATPase, you will be able to harness that energy to add a phosphate onto ADP, and that creates ATP. ATP generated this way uses what we call oxidative phosphorylation, because this whole thing is powered by oxidation reaction, right? What do I mean by that? To pump the H+ over, we need to use the high energy electron, right? Where did you get the electron? We got it from glucose. We oxidize glucose to get that electron, right? So this whole thing is powered by oxidation reaction, specifically oxidation of glucose, right? That's why this is oxidative phosphorylation. Just to recap it very quickly one more time. We've created a lot of energy carriers along the way. They capture high energy electrons from step 1, step 2, and step 3, namely glycolysis, pyruvate oxidation, and electron transport chain, right? We copy all those we capture all those electrons, and now we bring it to the ETC. ETC just refers to some membrane proteins that are found in the crystate, right? So when the NADH deposits the electron onto the ETC, it becomes empty again. It becomes NAD+. It can go back and pick up more electron. What do we do with this electron? The electron will get passed down the ETC from one protein to the next. And as it does that, the energy will be used to pump H+ from the matrix into the intermembrane space. Then you have a lot of H+ here in the intermembrane space. They really want to come back into the matrix, but they cannot because the inner membrane is blocking it, right? We learned in lecture 4 that the membrane does not allow charged particles to go through. So once they go, the only way to come back into the matrix is through this special protein called the ATPase, or sometimes it's called ATP synthase, depending on what you're reading. And as the H+ goes through the ATPase, it turns the enzyme, which then adds the phosphate onto the ADP, and then it creates a TP. Okay? ATP created this way is called oxidative phosphorylation. So each NADH carries electrons that have enough energy to pump enough H plus so that when they come back in, it will create 3 ATPs. We talked about that already, right? Now, what about FADH2? Okay. FADH2 has less energy. Okay, pay attention to what I have to say next. FADH2 has less energy. So when it drop off the electron, it's not going to drop it off at the first protein. It's going to drop it off later on, like on the second one. And because we drop off the electron later on, it's not going to be sliding down the ETC as much, right? So it only able to pump fewer H plus. And when those H plus come back into the matrix through the ATPase, you will only be able to create 2 ATP. Okay. So once again, the difference between the two is that NADH, you can drop it off earlier, higher up on the ETC, so you can pump more H plus. More H plus means more ATP, 3 ATPs to be precise. FADH2 has less energy, so you drop it off on the ETC at a later point, and you are only able to pump 2 H plus and create 2 ATP. Any question about any of these things that I've told you so far? Okay. Finally, what happens when the electron gets to the bottom of the ETC? Now, to help you understand this situation, I'm going to give you an analogy. Okay. So, when I take my kids to the park, okay, I tell them don't slide down until the kid at the bottom gets off, because you're gonna hurt the guy, right? But there's always 1 or 2 kids every now and then that stays at the bottom and they don't want to go away. Okay, so if that kid doesn't go away, the next one will not be able to slide down, right? So what do? Well, the parents have to come, okay? Maybe not so angry. The parents have to come and then say, hey, it's time to go. Okay, so the parent pull the kid away, allowing the next one to slide down the slide. That is what's happening in the ETC. When the electron gets to the bottom, it's not going to get off on its own. Okay, it needs the parent to come along and pull it off so that the next electron can slide down. So in this case, the parent in this analogy would be oxygen. Okay, oxygen. Oxygen will come at the end of the ETC. It will pull the electron off, and then it will become water. That's why oxygen is called the final electron acceptor. It accepts the electron, and that's the final destination, and then it becomes water. The oxygen is very important. Without the oxygen, the electron will not get off the ETC. If the electron does not get off the ETC, the next electron cannot slide down. If the next electron cannot slide down, we cannot pump H plus into the intermembrane space. Without H plus in the intermembrane space, they won't come back in to create ATP. No ATP, no energy, we die. Okay, that's why we need the oxygen, the final electron acceptor, to clear the ETC so that the flow of electron can continue. By continuing the flow of electron, I keep on generating H plus in the intermembrane space. They can keep coming back in. I can keep on making ATP, and we live. So that is what happens in the electron transport chain, the ETC. Any questions? Let's take a look at the PowerPoint slides now. Now this is basically the professional drawing. Okay, that's the ETC, electron transport chain, and the NADH will drop off the electron. Okay, never mind the H plus here. Electron will drop down the ETC much like a ball going down the stairs. The energy is used to create the ATP, right? Ultimately, the electron will be picked up by oxygen, and then you generate water. That's the picture we drew, right? We have the NADH dropping off the electron at the first protein that allows you to pump H plus into the intermembrane space. All this H plus wants to come back in, but the inner membrane is blocking it. So the only way to do so is to go through the ATP synthase. And when it does, we will create ATP. The electron at the bottom of the chain needs to be removed so that the next electron can slide down. That's the job of oxygen. What's missing in this diagram is FADH2. FADH2 will become FAD, right? And it will deposit the electron at a lower point. And because of that, it's not going to be able to pump as many H plus, and therefore it generate less ATP. So everything is written down here. Everything I told you, it's written down here. Just read these things, and you should be okay. Never mind about this this word here. Okay, you don't have to know that. So just read it through. Like I said, it's everything I told you already. Okay. Oxygen is the final electron acceptor because it can be oxidized into water. That's not true. Okay, to be oxidized is to lose electron. It's actually picking up electron, so it's reduced. Oxygen is the final electron acceptor because more electronegative than proteins in ETC. Okay, so that's something that I have to explain a little bit further for you. Electronegativity means attraction for electron. Okay, high electronegativity means high attraction. It really likes electron. That's what it means. Okay, if something has high electronegativity, it means it really, really likes electron. So we have 3 proteins here, right? 1, 2, 3 on the ETC. You will put the electron in the first one. In order for the electron to go to the next one, the next protein must have higher attraction for the electron, right? Otherwise, how do you pull it off? Right? So second one is going to have higher attraction for electron, higher electronegativity. And then the next one will also needs to have higher electronegativity, right? So highest electronegativity. This is lowest electronegativity, lowest attraction for electron. But in order for oxygen to pull the electron off, it must have even higher attraction for electron than these proteins. Otherwise, it's not going to be strong enough to pull it off. It's going to be stuck to this one. And so that is the correct answer. Okay, everything else is false. Okay, let's try this one. We haven't learned fermentation yet. We will in a bit. but just check all the other ones. Okay, so we wrote it down in the summary at the beginning of today's class. It is glycolysis and Krebs cycle, right? So questions like these are like possible on the quiz, right? And you really have to be able to like remember the facts, right? Like this is not something that you should look up on like from your notes, right? That's like something that you should be able to recall right away. If you have spent time looking it up, that's why it's not enough time, right, to complete the whole quiz. All right, so we're going to spend a little bit of time talking about disruption of oxidative phosphorylation. In other words, things that could interfere with ATP production at the ETC. There are a couple of common ways to do this. Blocking the ETC with certain kind of poison, inhibiting the ATPase, and making the membrane leaky. So let's take a look at each of these ones in more detail. Rotenone is an insecticide and pesticide. And what happens is it will bind to the first protein in the ETC and completely inhibits it. Carbon monoxide binds to hemoglobin and therefore not be able to transport oxygen. Okay, that's why we need to have a carbon monoxide detector at home. When you are exposed to carbon monoxide, your body does not your body does not deliver oxygen. Instead, it delivers carbon monoxide, which we cannot use. Without the oxygen, the ETC is not going to work, right? And cyanide binds to protein in the ETC and inhibits it. So there's a common theme here, right? Basically, what it means is whether it's the rotenone, whether it's the carbon monoxide, or whether it is the cyanide, they all disrupt the ETC by inhibiting the proteins. Once you inhibit the protein, it clogs the ETC. When the ETC is clogged, you cannot pump H+. That's the recurring theme, right? If you can't pump H+, the H+ is not going to come back in. You're not going to be able to make ATP. That's why these are poisonous substances. They interfere with the proteins in the ETC. Okay, so if the rotenone binds to the protein, inhibits it. Carbon monoxide, it prevents the oxygen from being delivered. Oxygen is not there, it's not pulling the electron off, ETC gets clogged up, and then same kind of problem with cyanide. Okay. DMP. DMP is a substance that is going to make holes in the inner membrane. Let me go back to this picture to show you. If there are holes in the inner membrane, then the H+ no longer need to go through the ATPase, right? They would just leak back through, right? Through the holes. And that's a problem because now we are drastically reducing the amount of ATP that's produced. The only way to produce ATP here is to it's for the H+ to go through the ATPase, right? But if we have the poison that makes the inner membrane leaky, then they just go back in without passing through the turbine, without passing through the ATP producing protein. So we reduce the amount of ATP that's available. And DMP was actually accidentally discovered as an agent for rapid weight loss. Long story short, some guy works in a factory that yields DMP for other purposes. He accidentally ingested some because his gloves were contaminated or something like that. And then for the next couple of days, he lost a lot of weight. Okay, and people thought this was the magic pill. for weight loss. But what they didn't know was they didn't understand how it was doing it. And what was happening is, like I told you, right? You make holes in the inner membrane. And it turns out that if you are on this pill, your body switched to starvation mode because you're not creating a lot of ATP, and your body starts to burn fat, right? Because it thinks you're starving. Problem with burning a lot of fats in a very short period of time is it can be dangerous. It could change your blood pH. It could cause you to, slip into a coma, and ultimately die in this case. And even though it was banned, but people can still get it from the internet in some like illegal places, I guess. And there are stories of people who purchase these things, trying to lose weight, not knowing the consequences, and people actually die from it, right? So there are some articles talking about, the father losing her daughter because of these poison, and he's trying to advocate for tighter control of these substances. Okay, so this is the different ways that it could disrupt it, right? So we talk about the rotenin already blocking the first protein, cyanide, carbon monoxide blocking the last protein, DNP making the membrane leaky, and oligomycin blocks the ATP synthase. So this accounts for all the ATPs that are produced, right, in each of the different steps. The only one that's a little bit different is the NADH that's produced in glycolysis. So let me actually go explain that to you. Okay, so maybe we go back to here for a second. There is this big table. Okay, so give you a moment to find that. All right, so let's try to track all the ATPs that are produced. Right, so starting with the first one here, glycolysis. How many ATPs are produced through substrate level phosphorylation? Well, hopefully you know by now it's 2 ATP, right?
Yeah.
Energy carrier produced is NADH, and we have 2 of them. Now, these 2 NADH is a little bit different than the rest. I'll tell you why. Let's go back to this note for a second. Okay. In glycolysis, where is glycolysis taking place in the cell? Do you remember?
In the cytoplasm.
That's right. Glycolysis happens in the cytoplasm, right? So when you make the NADH in the glycolysis, the 2 NADH, they are actually very big, and they cannot pass through the 2 membranes to go into the matrix. Okay, NADH is not able to go through. So instead of passing through the 2 layers of membrane, it will only pass the electron through. Okay, so something else has to pick it up in the matrix. Okay, and that would be FAD. Okay. So you might wonder, well, how come we don't use the same molecule to pick up the electron? Well, every time you do transaction that involves energy, some energy has to be lost. Okay, that's like heating a light bulb. You're gonna lose some energy as in the form of heat, right? So this is similar. When you pass the electron over, some energy is lost. So you're not gonna be able to pick it up with another NADH. Okay, you have to use something that has lower energy.
So that means in the matrix there is no NAD plus?
There is, but not for the purpose of glycolysis. Remember, pyruvate oxidation and the Krebs cycle happens in the matrix, right? So those processes will require NAD+. So those will be in the matrix. Okay. So going back to here, we create 2 NADH, which again, it's in the cytoplasm. So we have to transfer them to 2-FADH2 in the matrix because they cannot get into the matrix. They're too big. So how many ATPs are produced? Well, we're going to multiply this by 2, which gives me 4 ATP. Total number of ATP from glycolysis, either directly using SLP or indirectly using oxidative phosphorylation, is 6 ATP. For pyruvate oxidation, how many ATPs are we producing through SLP?
0.
0 is correct, none. Energy carrier produced from pyruvate oxidation, we have 2 NADH, and that's it. These are already in the matrix, so there is no need to transfer them. It's 2 NADH because each glucose creates 2 pyruvate, right? Each pyruvate will produce 1 NADH. That's why, it's 2, 1 from each pyruvate. We're going to multiply by 3, which gives me 6 ATP. 6 times 2, 3 times 2 is 6. So that's just 6 ATP. Krebs cycle. How many ATPs are produced through substrate level phosphorylation? Yes, Florence?
2.
2, that's right. 1 from each spin, right? We spin it twice for each glucose. Energy carrier produced, we have 6 NADH, 2 FADH2, right? They are already in the matrix, so we can take it to the ETC directly. 3 times 6 is 18. 2 times 2 is 4. (18) plus 4 plus 2 is going to be 24. 6 plus 6 plus 24, 36. Okay. That's how you get the 36 ATP. Any questions? All right. So just a couple more slides, and then we'll be done. Cellular respiration can use other kinds of molecules besides glucose. We're not going to go into a lot of details, but it's enough to know that fats and proteins, they can be broken down and be fed into the different steps of cellular respiration. For example, when you break down fat, and you might recall from lecture 2, fat is glycerol plus fatty acid. Right? So you can break it down into the fatty acids, and then the fatty acid can be fed into pyruvate oxidation directly. Okay, that's just an example. The proteins can be broken down into different amino acids, and depending on which of the 20 amino acid it is, you will be able to put that into different places in the cellular respiration as well. All right, so that's just a diagram that shows you, right? The fatty acid goes into can become acetyl-CoA, and then it feeds into pyruvate oxidation. The proteins can become different amino acid and feed into different parts of the Krebs cycle. Okay, so like glucose is the preferred source of energy, but it's not the only fuel source, right? We can burn fat, we can burn protein as well. All right, this is an example of an application question. You guys keep on asking me, right? Okay. I'll give you a moment to think about this. Okay, so I have a lot of questions. Some people are choosing D and choosing E when there are no D and no E. Yeah, don't do that on the test, okay? Like make sure you only choose things that are available. And maybe you choose D because you're not sure what the answer is, right? Okay, so let's take a look here, right? Hydrogen cyanide is poisonous. It binds to one of the membrane proteins in the ETC. We already learned that, and blocks the movement of electron. Okay, so here is my ETC. Electrons cannot move. Okay, it's just stuck here. They're not moving. If cyanide poisoning occur, what would happen to the pH of the intermembrane space? Okay, this is the logic you have to think through to answer a question like this. ETC is blocked, which means, are we pumping H+? Are we able to pump H+ over? Yes or no?
No.
No, right? This is all blocked, right? Okay, so you cannot pump H+ into here. You have less H+. Okay, less H+. Less H+ means higher pH or lower pH. According to the hint, low pH is caused by higher H+. So that means less H+ or lower H+ is going to cause a higher pH. Do you understand? On the test, if you see something similar, if it says hydrogen cyanide is poisonous, okay, and you remember, oh, we talked about this question in class, so you look it up, don't automatically just choose higher pH. Read the question carefully. I might, like the question on the quiz, I don't know which one you get on the test, right? I don't know which one you get, but it might say what would happen to the pH of the matrix instead, right? So in the matrix, because the H+ is not getting pumped over, so you have more H+ now. More H+ means lower pH, right? Okay. So you just because it looks similar to something we've done does not necessarily mean it's the same question, right? I think that's a mistake. Like I said, half of you may on this second quiz, because you put the cold environment answer when the question is asking for the hot environment. All right. Okay, we're gonna spend the last 10 minutes talking about fermentation. When does fermentation take place? In humans, fermentation occurs when oxygen demand exceeds oxygen supply. Okay, when you need more oxygen than you are getting, your cell switches to fermentation. The kind of fermentation we do in our cell is called lactic acid fermentation. Okay. Some people find it strange because when they hear the word ferment, they're thinking about, like making wine, letting your dough kind of ferment a little bit, right? To make it rise. That's another kind of fermentation. Okay. In our cells, when we don't have enough energy, oxygen, we switch to fermentation. An example of when that could happen is you're running at full speed, okay? After a minute or two, if you're running at full speed, right, you will start to feel tired, right? You won't be able to run at full speed indefinitely. So after maybe a minute or 2 minutes, the cells in your muscle is not getting enough oxygen. When it's not getting enough oxygen, it's not going to be able to produce enough ATP, right? Because not having enough oxygen clogs up your ETC, right? When your ETC is clogged up, let me go back to this screen for a second. When your ETC is clogged up with electrons, the NADH has nowhere to put the electrons. If they don't have anywhere to put the electrons, then you run out of NAD+, right? Because you're not converting it back into NAD+, right? Without NAD+, glycolysis, pyruvate oxidation, and Krebs cycle cannot take place because there isn't something to pick up the high energy electron. You have all these passengers waiting at location 1, 2, 3. We don't have the shuttle available to take them to the final destination. That's basically what's happening. So the purpose of fermentation is to regenerate NAD+. Okay, you're gonna take the pyruvate that you get from glucose, you're gonna break it down into lactic acid instead of sending it to pyruvate oxidation. And in the process, you regenerate NAD+. By regenerating NAD+, you will allow glycolysis to keep on happening. You're not getting a lot of energy from glycolysis, but still, you will be able to make 2 ATP. And that will allow you to keep on going for a little bit longer, okay? And eventually that will not be enough, and then you will have to stop running. In case you missed it, here are the key points. When you don't have enough oxygen supply, your ETC is clogged up. When the ETC is clogged up, you are not making enough ATP. You are not regenerating NAD+. Without NAD+, glycolysis does not happen. Krebs cycle does not happen. Everything stops. The purpose of fermentation is to provide an alternate path somewhere for the pyruvate to go, okay? Because it's not going to the ETC, it's not going to the Krebs cycle, everything is clogged up. So instead, it gets converted into lactic acid or lactate, which regenerates NAD+, that allows you to keep on going for glycolysis and produce 2 ATP. It's not a lot, but it's better than nothing. Okay, so that's just another view of what's going on. Fermentation in microorganisms. Okay, so things like yeast, like the one that we use to make bread, for example, they also do fermentation. Okay, but they do a different kind of fermentation. It's called alcoholic fermentation because it generates ethanol. Ethanol is the drinking alcohol. There are different kinds of alcohol, right? Like there's methanol, that's rubbing alcohol. There is isopropyl alcohol. That's the one you use to disinfect, right? So the one that you can drink is ethanol. You can't drink the other one. You drink methanol, you go blind and you die. Okay, terrible way to die. And then it creates carbon dioxide. So the purpose is the same, okay? Because the yeast, they don't have the complicated machineries to do Krebs cycle. They don't have the ETC, and they're very small, so they don't need, you know, the 36 ATP. They just need 2, and it's enough. So they take the pyruvate, and then they convert it into ethanol and carbon dioxide. If you're using the yeast to leaven your bread, to leaven your dough, that's because of the carbon dioxide. Okay, it causes it to rise. If you're using yeast to make alcohol, to make like red wine or something, so you mix the yeast with the grapes, let it ferment, it creates the ethanol, okay? And then it regenerates NAD+, allows glycolysis to keep on happening. Humans, we do lactic acid fermentation. For yeast, they do alcoholic fermentation. So the fate of pyruvate, where does it go? Generally, for us, when we have oxygen, and normally we do, okay, we're breathing just fine, then it's gonna do aerobic oxidation. That means in the presence of oxygen. You will go through the pyruvate oxidation, and then it will go to the citric acid cycle, Krebs cycle, and then etc. If you don't have enough oxygen and you're human, you will go switch to lactic acid fermentation, regenerating NAD+, so that glycolysis can keep on going. If you are yeast, right, you don't have the machinery to do step 2, step 3, and step 4. So instead, you create ethanol and carbon dioxide, and you regenerate NAD+. And that's it for lecture 5. All right. So please start studying. I will post the notes. I'll post the recording. I'll post the review package for lecture 1 as well. And we will do a review session on Monday, right? Next Monday. Okay. If you have other specific concern regarding your situation, feel free to email me. I will be sure to reply you. Okay. Thank you for coming. Again, don't feel discouraged. You can do it. Has been done before. People tend to be okay towards the end.
Sorry,